The following video shows a screen recording from the flexImaging software. It shows a MALDI imaging dataset of a mouse pancreatic tumor acquired on a solariX FTMS. After the MALDI imaging measurement, the same section was washed, then stained by H&E. A virtual microscopic slide of this stained section is then co-registered with the MALDI imaging data. A portion of the tissue was imaged at a spatial resolution of 50 micron. We can see from the gross anatomy that this imaged area encompasses several anatomical features, including a lymph node and portions of the tumor. A variety of mass filters can be selected, and the presence of that mass shows different distributions across the tissue. Selections of different masses show different distributions that can then be correlated to the anatomy. One mass marker in particular shows an unusual distribution across the tissue. If we look closely, we can see the presence of some darker areas that appear to be green dots. And the question can be asked, from what structure do these green dots arise? Once the image is co-registered, the flexImaging software allows for seamless fading between the mass spec signals and the virtual microscopy slide. If we overlay the virtual slide with the mass signals and zoom in, we begin to see additional detail in the tissue. From here, it becomes apparent that the green mass signal is always found in epithelial ducts. We need microscopic resolution in order to identify the specific arrangement of cells that form the epithelial ducts. Zooming in to a region containing ducts shows that the mass corresponding to the green signal is present in all of them. Additional correlation can be made through seamless fading between the mass signals and the microscopic image. At first sight, the global average spectrum does not appear to show many high-intensity signals. However, as we zoom in on the baseline, we begin to see the presence of many low-intensity signals. This is due to both the high mass resolving power and the high dynamic range of the FTMS instrument. Zooming in on a single nominal mass shows that it actually contains many different mass signals, which are very closely spaced. Frequently, these mass signals show different distributions across the tissue. In this example, we see 3 peaks separated in mass by less than 100 millidaltons, which show completely different distributions across the tissue, giving us ultimate specificity. For additional information, please visit www.bruker.com.